Okay, so my name is Dr. Rob Mayton from the University of Bedfordshire, and this short talk will take you through a review of current COVID diagnostics and focus on the future of LAMP testing in the context of COVID diagnostics and how and what the use cases for LAMP testing are. So let's start off with this overview slide. This is taken from Analytical and Bioanalytical Chemistry from a recent publication from this year, and this gives you a nice overview of current COVID diagnostics, and what we're going to do is step through this diagram and provide some explanation of the steps and look at what's going on at each step. So let's start off with sample collection, the first and perhaps in some respects most important stage of the process in many ways. So if we look at sampling, normally sampling will be by nasopharyngeal swab. It can also be potentially through things like saliva. Generally, you take the swab and it is placed in some sort of storage media. The storage media is there to try and keep the virus intact until we then go into the step of processing it for whatever analytical process we're using. And there are a variety of downstream steps available once we've got our sample into a media. One very important thing to think about at this stage is that most medias actually keep the virus particles intact to keep them preserved. And what then happens if we go through a lysis and extraction, when you get to the lab, the sample is lysed up, normally using a chaeotrope which breaks up the virus and releases the RNA from inside it. The RNA is bound to a column and purified away from the cell debris and viral debris, and then we take the purified RNA into a downstream reaction process. Most of these downstream reaction processes involve the generation of cDNA, a complementary DNA copy to the purified RNA using the enzyme reverse transcriptase. This is necessary because most of the downstream detection techniques are DNA based, not RNA based. So we have to convert the purified RNA from our virus into a DNA copy, complementary DNA, using reverse transcriptase before we can do any sort of detection. There are some extraction-free protocols around where you go straight from your sample to the reverse transcription test. This process can be made difficult because of all the junk that is in a conventional sample, and this can stop your downstream assay from working. So to get extraction-free protocols to work takes an amount of effort to make sure that you can then do the next steps in the process, the reverse transcription and whatever your downstream assay is. What we'll be talking about shortly is the other way that we test these things, through lateral flow assays, and in that case we take our sample stored in our media and take it straight again into an assay which looks at a slightly different thing. So we're going to look how we then first do things in lateral flow assays. Lateral flow assays are the most commonly used assay these days for general testing because they're a point of care test. So these tests are done with these devices which are familiar to people as pregnancy tests, where you put a sample on and then you look for the development of colour at 2 lines, a test line which says you've got the sample on there correctly, and a yes/no line to say, in this case, have you got COVID or don't you have COVID? So let's look at this in a little more detail. These things are based upon antigen-based assays. So what you're looking for is a viral antigen. This is normally one of the proteins in the viral capsid, which encases the virus when it is in its live, in quotes, infective state. The lysis buffer that you use will normally have a little bit of detergent in it to break up the virus and release the antigens from the surface, but this is not necessarily true in all cases. As long as you've got access to the viral antigen, which after all will be on the outside of the virus because it's the viral spike protein in many times, then you don't really need to lysis, but lysing the sample to some extent will increase efficacy as the number of antigens available will increase. What you have then on the line for your COVID test is a capture antibody, which is designed to bind to the antigen, and when you put your sample on in the sample chamber, along with the flow of virus down the thing, you will also have a detection antibody with a label. This will stick to another part of the antigen, and it will give us a signal. These are often a gold nanoparticle, which allows you to see whether you've got something bound or not on the assay. There is another type of lateral flow assay available, and this is through a molecular based technique rather than a protein based technique. So this depends upon first amplification of the viral genome using isothermal loop mediated amplification, LAMP, which I'll talk about a little bit later. So we amplify parts of the genome first, and then there's a clever bit of biotechnology using CRISPR-Cas that allows us to cut a single-stranded DNA probe which matches our target piece of viral DNA, and we release a quencher from a fluorophore, and we can see a readout on our lateral flow device. This could be again a gold nanoparticle to allow us to look at it in exactly the same way as the other one. The difference being we're detecting, instead of a viral protein, we're detecting the viral RNA amplified into DNA, and then used a detection mechanism to allow this to be picked up and give us a visual readout. So we've talked through how we collect sample and the two lateral flow based assays, the antigen based assays, which are in huge use these days, and the lateral flow CRISPR-Cas based ones using a amplification. The next step is to talk about how the majority of testing was done in the beginning through qPCR, quantitative PCR. So if you remember, the first thing we've done is to get our RNA and copied it into cDNA, and then we use a PCR based reaction to amplify it. In terms of swab testing that is done in labs, this virtually all has to go through an RNA extraction process, because the qPCR reaction is not tolerant of the sorts of junk that we get in our swab samples. So we have to extract our RNA and purify it, because our reaction is not tolerant of the junk that we have in our original sample, because the junk stops the reactions from working. So to do PCR, what we're going to do is then amplify our copied DNA and detect it. So let's look at these steps. So we copy our RNA into cDNA through reverse transcriptase. We then amplify this through a polymerase chain reaction. This uses forward and reverse primers to amplify a specific part of the viral RNA copied into DNA. We normally, in this reaction, use a third probe primer, and this probe will release a fluorophore as part of the copying process, which allows detection. And what we do is, as the fluorophore is released, we see an increase in fluorescence, which is proportional to the amount of DNA copied. What we then measure with a positive or negative is we measure the number of cycles, a cycle time, the number of cycles needed to cross a threshold level of fluorescence, and we say how many cycles has that taken. In terms of qPCR, we can theoretically detect down to a single molecule, which would take around 37 cycles. Anything beyond that is not considered to be a reasonable number. If you are highly positive for COVID, you will have a CT of around 20 to 25, and that's what most infectious people have. The other part of what's being done alongside this is sequencing, and sequencing is what allows us to detect variations in the viral RNA, which are related to the different types you see when you hear people talking about things like the Kent variant or the Indian variant. Then this is talking about mutations present in the RNA. These are sequenced by copying the whole genome. So from our PCR amplification reaction, we create a range of DNA fragments which cover the entire genome of the virus. These are then sequenced by two different sequencing methods, either sequencing by synthesis, or we can do whole genome sequencing using nanopore technologies. I'm not going to talk about this anymore, but these two mechanisms allow all of the bases of the genome to be read. Lastly, we're going to move on to LAMP, loop-mediated amplification. This, again, has to take the first step of reverse transcription from whatever sample we have. Now, LAMP uses a polymerase which is more tolerant of some of the junk that we've got in our sample, so we can use extraction-free protocols more readily. Although there are a number of LAMP reactions which will use purified RNA, which gives you some advantage because you concentrate up the RNA before you detect it. We then still have to turn it into cDNA. The loop-mediated amplification then allows a colorimetric readout. This is because as we amplify our DNA copy of the viral genome, it changes the pH of the solution, and we have a pH indicator. If there's no change in pH, then it stays red. If there is a change in pH, it goes yellow. This is actually one of the things which is considered to be limiting for LAMP, because many people consider LAMP to just be a yes-no answer, rather than in terms of the amplification that we saw previously using PCR, when we actually get a quantity of virus. So one of the advantages of qPCR is it will actually tell us how much virus there is in a cycle, and is both considered to be more sensitive, but also gives you an important value, the CT value, that tells you how much virus somebody had in them. And this can be linked to whether they're considered to be infective or infected. In other words, if they're infective, they can pass on the virus to somebody else. If they're infected, they may still show traces of virus in their system, but not actually have any infective virus left, which could infect other people. It is known that most people will get rid of virus after around 10 days to 2 weeks after they've first been infected, but they may stay positive to qPCR for up to 3 to 4 weeks, which can be a disadvantage because you're saying you're still positive for virus in your system. This is not actually active virus, this is remnants of virus from you clearing up your infection. Now what we'd like to do is turn LAMP, which is an easier method to use, into something which gives us a more accurate readout like qPCR, and this would allow us to use LAMP to give a more accurate and sensitive determination of not just have you got virus, yes, no, but also some indication of viral load. So that's a walk through the different sampling techniques and the different analytical techniques that are available. So we're now going to talk a little bit more about LAMP in detail. So LAMP is loop-mediated amplification. This is a DNA-based detecting molecular tactic. This is a DNA-based LAMP is a DNA-based detecting molecular technique similar to PCR. It uses more primers than PCR. As you've seen in the previous slide, PCR uses 2 or 3 primers, 2 for amplification, 1 for detection in most PCR assays. With LAMP we use a minimum of 4 and normally around 6 primers to amplify our DNA target. And this uses what's called a strand displacing polymerase that allows isothermal amplification of a dumbbell product. That's shown in this diagram here. You lyse up your SARS-CoV-2, you release the genome which you copy into DNA, and then you have a set of primers which target specific areas, normally the E gene, the N gene or ORF1, and these will amplify up this piece of DNA by producing these dumbbell shaped structures where the primers loop around and allow copying of a double stranded fragment into a high molecular weight amplicon. Because it's isothermal, it doesn't require the heating and cooling steps that are necessary for qPCR. This makes it faster than PCR, And it has a simple detection mechanism by pH, because as you amplify your DNA target, every nucleotide you add releases a hydrogen ion into the solution, which makes it more acidic. You can therefore measure a positive result by a change in colour from a red colour at the normal alkaline pH to a yellow colour at the acidic pH that you get after you've made lots and lots of product. At the same time, you can also monitor this reaction by an alternate colorimetric method which measures the amount of free magnesium in the solution, which again changes as part of the amplification reaction. The problem with LAMP, it is considered to be less accurate. There are some issues with the background signal because there's a tendency for these primers to produce some sort of product, even if it's not the correct product, if you leave them for long enough in a reaction. So if you let your reaction go for long enough, it will often start to change colour. And it's perceived also as being not quantitative. This is from the NEB website, New England Biolabs. They're one of the biggest manufacturers of LAMP technology kits. And this shows you their slide about using LAMP for detection of COVID. And what you can see is you get this very typical yes, no answer. However, in reality, even if you measure pH change, what you will see is it's not a yellow or not yellow. You get a graduation of results, and this is a dilution series. This is again from the NEB website, where you can see at high concentrations you get a very distinct yellow colour, and then it fades towards pinkish and more red as you use less and less target. So actually you can see from this that LAMP is capable of being semi-quantitative at least. So what are the problems and solutions with the use of LAMP as an alternative to qPCR? Well, qPCR needs expensive equipment and a lab to set up your reactions. You need this purification step for virtually all qPCR reactions, and you need an expensive machine to do the amplification and detection of your product. And LAMP has big advantages because it will allow point of care testing using suitable equipment, meaning you can actually test things where people are and give them an answer very quickly. So the advantage of it being more tolerant means that we can do direct testing of people's samples without the purification step and hopefully produce answers in less than half an hour rather than the couple of hours it takes for a typical qPCR assay. LAMP is perceived as being qualitative and hence less accurate. Well, the answer is it's not necessarily entirely yes, no. If we use continuous optical monitoring of the reaction as for qPCR, we can actually go from being yes, no to watching the colour change over time to get some idea of relative amounts of viral load. There is the potential of using fluorescence as a technique for detecting LAMP. However, the problem with fluorescence is that takes us back to needing more expensive equipment, which takes away one of the prime advantages of using LAMP, of cheaper and simpler equipment. We talked a little bit about the background issues that LAMP has. There are a couple of answers to this. We use improved primers, which are less likely to generate background, and there have been a number of research articles pointing to how we can do this. And also we couple up a more intelligent machine learning in terms of analysing the sorts of signals we get out to allow us to analyse our signal and come up with a better answer. Obviously fluorescence takes us back to another way that we can potentially get a better answer from this. LAMP has a lower detection limit. It's generally considered to be around 10-fold less sensitive. That's the equivalent of around 3 CT in the number of copies it takes to detect a reaction in our qPCR. The qPCR limit, as I mentioned previously, is considered to be 37. Now actually there's a lot of evidence suggesting that people who are infectious actually have CT values which are less than 30. So actually, is this a problem? Depends upon what question you wish to ask. If you want to say, is somebody infectious, then it's likely that LAMP is easily going to be sensitive enough. your loss of around 3T in comparative sensitivity will still give you a detection limit which is significantly above the level where people are considered to be infectious. However, because the PCR limit for because the PCR limit for qPCR is down to single molecules, and this will give you positives for people who have been infected but are no longer infectious, you will always get a false negative for some samples where the lower detection limit of LAMP will say this person is negative, but for qPCR they're positive. However, even though they are a, in quotes, false negative, they're actually highly unlikely to be infectious. In terms of a comparison to lateral flow, the sensitivity for lateral flow is a very interesting question. Remember that the lateral flow assays, the prime ones that are being used, detect viral proteins. Now, viral proteins are an indicator that you've got generally live infectious virus around. Now, this means there are two potential reasons why lateral flow may not pick up your sample: lack of sensitivity, but more importantly, just lack of actual complete virus. And this goes back to the issue with qPCR, where qPCR will detect fragments of virus, not infectious virus. So lateral flow may not be as bad as people say, because actually it's probably very good at picking up infectious people, and many of the false negatives are actually people who are no longer infectious but have been infected recently.